NADPH as a co-product. In this reaction, NADPH produces the oxidized glutathione and turns into an ADP positive. The fifth and last mechanism of how antioxidants protect us against free radicals, which I'll cover in this video, is the activation of sirtuins. Resveratrol will be our example of an antioxidant.
The last mechanism.
Resveratrol is known to activate a class of enzymes called sirtuins, particularly sirtuin 1. By promoting the expression of antioxidant genes and proteins, sirtuin 1 activation helps to counteract oxidative stress and protect cells from harmful reactive oxygen species. Additionally, sirtuin 1 is involved in DNA repair pathways, contributing to the maintenance of genomic stability. Activation of sirtuin 1 can enhance cellular resilience against oxidative damage and support longevity. As a quick recap, the main mechanisms of how antioxidants protect us against free radicals discussed in this video were hydrogen atom donation, electron donation, metal ion chelation, antioxidant enzyme regulation, and the activation of sirtuins.
Okay. All right, so there is a 5. Let me share the slide first. Maybe hybrid. So here I show the summary just now. So the video will explain actually each of the molecules, what happened on the first mechanisms, the second mechanisms, the third mechanism until the fifth mechanisms. So in your research, in your final year project, in your S2 or S3 project, anyone here involved with the antioxidants? Anyone here? So now, did you get any idea which antioxidant mechanism you belong in your research? Ada idea? Kan masih pening-pening. So now you know that antioxidant is a general terms. So when you go for your supervisor, inform them you get the antioxidant results, make sure that you can explain what happened actually, what activity inside your results that you get. So which mechanisms you belong, you have to know which one. So that is a 5 mechanism, the major one. So which one is you? Normally for my part, I will belong onto the mechanism 1 and 2 for my part, for my field of interest. So I came here, I just give an overview. So you have to choose which one is yours. I just give an idea. So antioxidant is a general. So don't give your supervisor is just mean value, standard deviation value. And when your supervisor asking you what happened to the activity, so you have to know to answer based on the mechanisms, the 5 mechanisms, which one is belongs to you. All right? Okay. So after you know that, you know which mechanism that you belong, then you came to the measurement or test or analysis antioxidants activity. Okay, normally ada FREP, DPPH, ABTAs. So, mana you pilih? Suka hati. Normally my student suka hati, yang mana ada? Yang mana ada available, okay, I do FREP. Kalau FREP tiada, ABTS. And then, satu lagi, DPPH. So when I ask, kenapa you pilih DPPH? Why you choose DPPH? Then diam. Then the answer is, sebab chemical itu sahaja available, doktor. Then nak je tarik telinga. So bila thesis, you defense your thesis, you defense your proposal. Kalau itu jawapan yang you bagi, what happened to your marks? What happened to your defense? So you have to know, kenapa you pilih DPPH? You have to know, kenapa you pilih this technique? So I list out here, measurements of the antioxidants activity. Kenapa? Ada. It's okay, it's okay. Ada sedikit lagi. Alright. It's okay. So, first, a radical scavenging method. So if you focus on the ROS, you want to scavenge. Scavenging is the tangkap, radicals, free radicals, you want to tangkap. Okay, so if you want to tangkap, you can use the oxygen radical absorbance capacity assay. If you want to focus for the ROS, there is 4 methods available. The first one, oxygen radical absorbance capacity. The second one will be chemiluminescence assay. The third one, DPPH. The fourth one, Trolox. So kalau you pilih DPPH, you will belong to scavenge the ROS. So meaning that you will come to the first mechanism just now, the ROS. Okay, understand? If you focus on the non-radical redox, for example, you use a FRAP. So is it applicable? Bolehkah kita guna? Kalau tiada chemical, kita guna DPPH. Kalau ada chemical, kita guna FRAP. So boleh? Is it boleh? So betul antioxidant, which one that you focus? What type of antioxidant you want to, mechanism of the antioxidant you want to focus? So if you want to focus the DPPH, meaning that the free radicals, you want to study the free radicals. If you use the PrEP, you focus on the non-radical redox potential based methods. So it's a different techniques and different yield that you will have. That you will get a different yield and then different mechanism, all is different. So tidak boleh, saya tidak boleh terima, for my student, I tidak boleh terima jawapan antioksidan apa you nak buat. Ada DPPH saja doktor? FREP tiada. Then you focus on which one? You said to me previously you want to focus on the ROS. But suddenly you pergi FREP. And your study from the first objective until the last objective belongs to the ROS. So why suddenly you pergi FREP? Kadang-kadang I marah juga. Okay. So the next one, metal selations capacity. This one, when we study about the heavy metals detections, and then we are using the extract from the plant. We extract the phytochemical, and we want to detect the heavy metal inside the fish or seafood product. I have to study that one. I have 2 students that doing that one. So the antioxidants will focus. They're using an antioxidants analysis based on the metal selations capacity. Okay, the last one is TPC. Yang ini paling happening, TPC, total phenolic compound. Okay, when we talk about total phenolic compound, I told you just now this one. If you focus on the phenols, okay, TPC, you will run the TPC. However, my next questions, which phenolic compound that you focus? There is a lot of phenolic compound. There's a lot. So, which activity that you want to focus the antioxidant? Kalau for S1, maybe I boleh terima. S2, at least you know a little bit. For S3, you have to know the specific of flavonoids that you extract. For example, okay, doctor, I want to focus on the flavonoid. My next question is, which flavonoid activity you want to focus? You have to know. Sampai dalam-dalam tu. So, maybe you will tell me, you want to see the differences between these 5 flavonoids. That one is for S3. Okay, so that is a different level, S1, S2, S3. So that is a different level actually how we analyze the words of antioxidants. Bukannya bagi saya the number of the mean and number of the standard deviation. Tiba-tiba bagi saya yang itu, and then I ask, is there any significant difference? I don't know lah, doktor. Maybe doktor yang kena buat sendiri ya. I kena buat sendiri kot, I kena buat significant. I think for S1, you have to know the significant difference between the value. Kenapa you buat SD? Kenapa you buat mean? What is the meaning of the standard deviation? Bukan suka-suka sahaja itu ada di situ. Okay, so we can see the error also from there, right? Kalau S3 kena tahu what is the compound phytochemical that you focus. Bukan flavonoid, you kena bagi lagi dalam. Alright. Kalau S2 I boleh terima sampai flavonoid, phenolic acid itu boleh. Kalau S1 I boleh terima sampai phenol TPC. Alright. So seterusnya, this is the radical the Ross scavenging method. The first one, oleh accumulations. Okay, kita ada dua. The blue one is the ROS. The second one is the redox potentials assay. So if you click here, you will go directly to the link of the pembuat method there. Okay, you boleh follow. You tekan link this one, for example here. Situ keluar di ya. So tempat I tak keluar slide saja. So you boleh klik terus Okay, if let's say suddenly you have an idea now, tiba-tiba ada idea, you focus on the antioxidants, suddenly you think that you focus on the DPPH, you suka DPPH My student sangat suka di situ, DPPH, FREP, dua benda ni dia suka. So kalau you focus on FREP, you boleh fokus kepada Molina. Molina is the here. Bolehlah. Kenapa tempat saya ni? Okay. All right. So the next one, antioxidant evaluations in food model system. Let's say you ada buat product, you develop a product from the maybe krepek. Okay, maybe you produce a product from the seafood. Okay, become a product, gula-gula ke, snack ke. Okay, so this is the measurements model in food system. First, you can focus on the measurements of antioxidant activity in the oil system. Let's say you want to focus on the krepek. You know krepek, right? Chips, krepek chips, krepek ikan, keropok, keropok ikan. Kalau sana krepek, krepek, keropok ikan. Kalau you want to see you want to produce a keropok ikan. So maybe you want to see the antioxidant activity in the oil system. Kita makan keropok, kalau ada keropok tu kita boleh find the antioxidant activity in oil systems by using the oxygen consumptions. Okay, by find out what is the oxygen consumptions, and then calculated the change in oxidation substrates. Also, you may measure the oxidation products as well as the system change during the oxidation process. So ada banyak jenis kita boleh. So ada 4 results. Just antioxidant. Okay, trust me, just the results of antioxidants you can publish. Tapi bukan Q1. Just maybe Q3, Q4. Tapi just the results of antioxidant. As long as you do the analysis and then analyze, the words of analyzing the result. So the words of antioxidant just now, it will give you 4 results. Oxygen consumptions. Okay, you calculate the changes in oxidation substrates. You can calculate the oxidation products. And there is a 4 mechanisms here that you can report. Right? Normally, I will get for the antioxidants results. Normally, I don't know here. Normally, as a lecturer, normally my student will give me just